Previously, we have understand how this CAR T cell therapy works. More specifically, we have understand how the second generation CAR T cell therapy works. In this video, we will cover what are the side effects of this CAR T cell therapy and what are the possible solutions that are presently been studied. So, what happens when the CAR T cell works is that it will also trigger uh, it will also trigger the other immune response that might uh, overstimulate and into the cause problems to the host. For example, this activation of CAR T cell will leads to the release of this GM-CSF, and that will finally leads to this to either neurotoxicity or CRS, that is cytokine release syndrome, or both of them. And along with that, along with this CSF pathway, it will also leads to activate this macrophage, and this macrophage will cause increase will cause the will leads to the increase of this interleukin 6 and interleukin 1, and this interleukin 6 and interleukin 1 will further auto activates this macrophage. So this uh, cycle will go into the loop whenever the CAR T cell is in function, and this will cause not only the neurotoxicity but also the CRS, this cytokine release syndrome. So, and this self-activation, self-amplifying inflammatory activation will leads to create many problems and um, multi-organ defect. Because of this, neurotoxicity and CRS might even be lethal in some cases. So, what are the present approaches that have been studied? So, before moving on that, one thing that we need to know is that uh, till now, the process by which this neurotoxicity gets activated is still not known in terms of whenever the CAR T cell gets affected, how this neurotoxicity gets activated. So now let's come back to the approaches that are presently being studied. One, the basic idea to prevent this kind of self-amplifying inflammation activation is to break the loop. So one of the approaches that people are using to break the loop is by introducing this uh, Anakin, Anakinra. So what it will does is it will inhibit, or it will prevent this interleukin 1 to further activate the macrophages. And another similar kind of alternative is this Tocilizumab. So what it will does is it will, instead of interleukin 1, it will work on interleukin 6, and this Metyrosine, or it will prevent the self activation of this macrophage into the host. So these are the approaches that are targeted towards macrophages pathway. Another pathway that is targeted is via preventing this cell signaling pathway through this uh, preventing this signaling pathway towards this neurotoxicity and CRS via inhibiting this GM-CSF. So this can be done with the help of this Lenzilumab. So it will provide the antibodies that will inhibit this GM-CSF to further signals towards the activation of this neurotoxicity and cytokine release syndrome. And one of the approaches that are presently being used is by presently under can be used in the clinical trial is by controlling the level of CAR T cells. So in this case, what happens is that whenever there is a symptom of this CRS is observed in the patient, this CAR T cells function can be suppressed with the help of this uh, Dasatinib. So what it will do is it will prevent the signaling of these CAR T cells to activate the immune response. So whenever there is a CRS, symptoms of CRS within the patient, this Dasatinib can be injected, and it will function as to suppress these CAR T cells instead of killing the CAR T cells. So in this case, uh, the CAR T cells get inactivated. So and whenever the level of this CRS goes below the threshold, or goes below the threshold level, yeah, at that time this CAR T cell therapy can be reactivated through the injection of this CAR T cells, and in this way this CAR T cell therapy can be carried out on the host. So these are some of the approaches that have been presently been studied to treat the patient through this CAR T cell therapy, at the same time controlling these side effects that are formed because of this CAR T cell therapy. And another problem that people are facing to understand these side effects is because there is not a perfect model available in the mouse, immunogenic mouse, uh, from where in which they can study this kind of side effects in detail. So, and the studies are also been going on in that area to create a mouse that can replicates the not only the tumors, immunogenic tumors, but also the this self-amplifying inflammation activations similar to the human. And if you need to understand more about the side effects of CAR T cell therapy, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.